I'm Emily Miller, and I'm a physician scientist at Albert Einstein College of Medicine. I'm studying coronaviruses, not just things like SARS-CoV-2, the virus that causes COVID-19, but also coronaviruses that are currently existing in animal populations that might one day become a human health problem. We're trying to develop strategies that would be broadly active against a wide range of coronaviruses, so that should tomorrow one of these viruses pop into the human population, we would have the toolbox of therapeutics and vaccines to tackle the viruses right from day one. So we know coronaviruses have a protein on a surface called the spike. And this is the protein that the virus needs to use in order to get into your cell and cause an infection. And vaccines, antibodies, these all target the spike protein. But the spike is actually composed of two different subunits. So let me show you what the coronavirus spike looks like and how it gets into cells. So this is a picture of the spike. And in red, you have the S1 subunit, and in yellow, you have S2. And it's hanging out on the surface of the virus. And then it comes in contact with your cell and it binds its receptor ACE2, which is present on the surface of human cells. And this interaction allows S1 to sort of fall away, exposing S2, and then allows S2 to spring into action. So S2 extends these arms. Then it undergoes this really interesting conformational change where its arms fold back on each other and it tugs down the viral membrane and brings it in contact with the host cell. And this creates a pore or a hole between the virus and the host cell and allows for the viral genome to go into your cell. So our current vaccines target the spike protein, but most of the antibodies that they stimulate are targeting the S1 subunit because it's what's exposed to your immune system. And the problem with that is as the virus sees your immune system, it mutates. And so the antibodies that you're making are now no longer the best antibodies against new variants that arise. And so this is why we have to update our COVID shots every year. But S2, because it's hidden underneath S1, hidden from all this immune pressure, it's more conserved among all of these different coronaviruses. So if you could incorporate an S2 targeting component of the vaccine, you could potentially have a vaccine that would be active not just against future variants of SARS-CoV-2, and then also novel coronaviruses that may one day jump into the human population. So I did my MD PhD here at Albert Einstein College of Medicine. I worked in the lab of Dr. Kartik Chandran, and we studied Ebola virus and how it gets into your cells. He was a wonderful mentor. He taught me a lot. And then I went off and did all my clinical training, became an infectious disease doctor, and was really interested about getting back to my virology roots, but wanting to bring all of my clinical knowledge and experience that I had gained from my clinical training to the work. We had stayed in touch all these years, and he brought up the idea of, wow, wouldn't it be fun if you came back here and you worked with me and we tackled some of these questions that you're interested in. And through that, it became this wonderful opportunity to sort of come home again to Einstein, this time as faculty. I think being a physician scientist is really the greatest job one can have. I might be a little biased, but it's so fun to be able to be in lab. It's such a gift to be able to come in every day and try to answer and ask important questions that I think will really impact human health. But then to also be able to be in the hospital and take care of patients, it keeps you focused and grounded. It helps you ask the right questions because you see actually what is important for patients. What are the things we really need to be dealing with? And so being able to do both and bring both together is really exciting. It's incredibly rewarding. And it's just the greatest career that one could have.